Now let's talk about some things that will be useful when you are making your workflow. A little bit more about the user interface and what features you will find there. Some actually very important. So let's begin with basic pipeline organization tools. Very basic one, graph annotations, then compartments, which are more involved and very important, groups. Then I talk about how you share your workflows, and then other useful features that you will find here and there. So you can annotate your pipeline. These are colorful pipeline annotations. These can be found here in the top bar of the user interface. And there you have like a text box, you have like a group box, arrow, and an image. And I have here an example of how they look like by default, and you can configure them to look different. So, for example, if I go in here, then I can draw a text box, this icon here, the T. You can draw a outline, then type in some text that you want. You can right-click or press Escape, then it will go back to the standard tool. And these you can move around, and they have a lot of settings. When you go to the parameters, you can change the text. Of course, you can put the content, the small text below. You can change the colors. You can align them. Then there's a group box. It's just a variant of this text box that has the text a bit differently aligned. This is intended for if you have like a set of nodes. For example, here this is something that will handle the glomeruli. So, okay, I write glomeruli analysis. Then it's always displayed behind the actual workflow. And these you can also modify. What my colleagues usually do is make them a bit brighter and nicer. So, for example, it's outline, make it a bit thicker so you can visually identify these easier. You can always edit them and change their size, unlike the standard nodes. Then you have also arrows if you want to show something. And then you can also embed images. So simple, small images you can do with this image tool. Now we get into the interesting part, because this is talking about compartments. So what are graph compartments? Graph compartments are a way for organizing larger pipelines. So this is one of the big disadvantages of visual programming. These pipelines can grow to impressive sizes. Real-world pipelines have at least 100 nodes usually. And you can imagine that over time, this small pipeline, they will look like that at some point. They look like you threw noodles at the wall and then you can't see anything anymore. And this is why JIPipe has a primary way of organizing your pipelines. It's called compartments. You've seen always this tab open. It always opens by default. If it's closed, you can access it via this top bar. If you visit this compartments tab, it looks very similar to the pipeline that you have edited already. It also has an Add Notes panel, it has bookmarks and history and everything. On the left side looks very, very similar, but it is something different. It is something that is above these pipelines where you do actually your analysis, structure of files, make your visualizations. This is a purely structural pipeline. And you see on the Add Notes panel that it doesn't have any functional nodes. It has no import image. It has basically only these annotations, because they are also just nodes, and graph compartments, they're called. So what are these? Why do you have this editor? This allows you to basically make a pipeline of a pipeline. Your top structure of how your pipeline is organized. You can imagine you have one that is pre-processing, analysis, and then post-processing. And these contain actually the nodes that do the work. You can double-click them, they will go in. We will soon make custom ones as well, and then I show you how to do this, so you can really manage these hundreds of nodes in a feasible way. Let's structure our project. So we are at this current state. We have some basic structure here already, but you can imagine if this is a larger pipeline, at some point this will be super, super large. So let's do a small structuring. For that, we go to the Compartments tab. You can also always click this button here. It will then jump there to this tab when it's not there. This looks like that. This is the default structure that you get always when you start a new pipeline. Again, here's a quick guide how to navigate through it, how to create new things and what it is. We go here into the compartments overview. So what I did here, that's why this panel is if you want this, you can keep this right panel always on, then it doesn't jump around. Or sometimes when you deselect things, it jumps. And then currently we have one compartment. The pipelines are always having an organization into compartments, but by default, JIPipe will make one for you that it will just jump into for you automatically, called analysis. But we want basically everything that handles the files, and here in our case, maybe also the importing and the basic pre-processing be separated off into its own compartment, and then have the tissue analysis and the glomerulus analysis separate. And then now we're going to add 2 additional compartments, one for the tissue analysis and then one for the glomerular analysis. Just put them in as any node, drag them in, and then it asks you for a name. So I'm going to call this a tissue, and then I make another one that's called glomeruli. You can double click those and they will open up in separate tabs. And you see they are empty because they are basically separated off from the other parts through this compartment system. When you double click them, then you have 3 tabs, each corresponding to the analysis, currently called still analysis, one called tissue, and then one called glomeruli. And everything is still in here. So the others are empty. So we're going to organize things now. And for that, let's move out already the nodes into the compartments where they should go in the end. So this analysis compartment that we are already in, it will stay. It will keep some of these nodes around, but only those for everything that has file handling, loading of the image, and our pre-processing, because it's such low effort pre-processing only media and filter. But this part here, this is for our tissue analysis. And this part here, this will be for our glomerular analysis, and we move these then out of the pipeline. So let's cut off the part that starts with this first outer threshold 2D. Basically the one that targets only the tissue. And then we slice this now out of the pipeline. So you can right click and cut it out, or you can copy it if you want to be careful. So I'm copying it. And then I'm going into the tissue compartment. And there I'm going to paste them in. So you see some of the connections got broken because they're literally not there anymore. I basically sliced this off, but we will fix this later on. So this is then our tissue compartment. These you don't have to replicate this. I just mark them as basically deleted. And then I'm going to take everything that handles the glomeruli. I do the same thing. I copy them out. I put them into the glomeruli compartment, paste them in, and then this looks like that. It's again disconnected. Convert RAW to RGB is also disconnected from the original image. So I'm going to delete them now. Delete. So now they sit there. Our analysis compartment that's still called analysis, it still works because you can go up to the media and filter. Everything's still there. But everything else is disconnected and broken. So if I go to tissue and then I want to run my visualization, it has no incoming stats because it got disconnected while cutting it up. So we have to reconnect it, and we do reconnect it systematically. Because you see, I don't see my other nodes here. They are only the ones that I put in here. We need something to interface between them. And for that, we need to make outputs, how they are called. We go into the compartments graph again. You see here that the one that I loaded in, it has already an output added. This is just historically because JIPipe compartments always had an output, but then now they don't. The new ones they have no outputs yet. So it's basically you can have like a box, something happens in there, but what stays in there stays in the box, basically. Unless you tell JIPipe what goes in and out and which ones are connected. So basically you have to put connectors in the pipes like with other nodes. So if your analysis one doesn't have yet an output, then you add one. I'm going to delete this one so I can showcase it to you. This analysis, first of all, it's not the analysis anymore. It's like any node, it has parameters here on the right-hand side that you can change. I'm calling this file handling. You can call it anything that you want. And then we have the following structure. And then we are going to add outputs. So we are going to make output interfaces, a special kind of node that basically allows you to pass data between compartments. So for the file handling, you only need to add one. So you need to only add one. You click the big plus button in the middle, leave the default name. It doesn't matter. You can name it as anything as it allows, but I leave it as output. And then when you go into the file handling compartment and edit this output here, as you've seen, then you notice that there is a new node that appeared. Maybe here it appeared on the top left. You can move it around. Maybe it's also behind some other node. Just move them. It's somewhere. And it's called output. You see that it has a plus from both sides. This means it's like a node that where you can customize the inputs and outputs. You can add as many as you want. And this node will be there for us to pass the data between compartments. Where will the data go? That is output here in our file handling compartment. You go back and then we just make a connection like with any other graph. I want my file handling outputs, what is produced there, available in tissue and also available in glomeruli. And when you do this and you go into file handling, you notice that now that it has an output connected, it indicates it that, okay, this is something that goes out of your view. So what we're going to output through our interface? First of all, I want my raw image, the original one, being out. So I move this a bit so you don't get confused with the overlaps. I want my import image out. Just drag a connection to the input, drag it in there, it snaps into place, into the plus, and then it will ask you, I call this one raw because it's the raw image. You can also give a description that says, okay, this is the raw image. And then I add it in, and then you see that it has both raw as input and output. This is a purely structural node, basically, that flows where something goes in and then it just goes out again. So what we also want out is the preprocessed image, because this is the one we want to do our analysis on. I drag it again to the plus and call it preprocessed. You can also create the slots here manually by clicking the plus, but this drag and drop is usually very useful because it auto-selects the correct type. Because you have seen here, there's a lot of types on the left-hand side. This is everything that JIPipe can work on, and then you have to hunt down the type, and this is quite annoying. This output here, it's just a node. You can run it. For example, I can update cache it into the slot raw. There goes my raw image, and into the preprocessed one, there goes the preprocessed image where you have the median filter. Because we made the connections already, the data is now available as well in the compartments that we have connected to the file handling. We will see this in a minute. When I go to compartments, and then, for example, I open the tissue. You see a new node appeared here. This is the same output. This is exactly this one. These are the same nodes, but this one comes from above, and this is there because in compartments we made a connection. If you disconnect it, then it will disappear. This is the way how you pass information between those compartments. It also says here in the name that this is the output from file handling, where it comes from. And also, as a nice hint, you can double-click them, then they will jump into the compartment where they originally come from. And then when you double-click them here, then you can jump quickly to the targets of that one. So I can go back to tissue. And then let's reconnect it. So because we broke all the connections initially. So here's our auto-thresholding. It was thresholded originally on the pre-processed image. So we are going to reconnect this one. And then on what was the converter to RGB made, it was drawn on the raw image, so not the pre-processed one. That's why I moved it a bit over. And then I am moving here the converter to RGB to the left. And then this is basically functional again. I can run here my converter to RGB, it's back. Everything works again. And then let's do the same thing to the glomeruli. We go into the glomeruli compartment. There is again the file handling output that has our data. I make some space and move it around. And then we are working on the pre-processed image again. And then the converter to RGB again has the order in a way that is maybe not optimal. This time I show it with the slots panel. You can also right-click it and move it. And then it's reconnected. And it works again. So, and then we have our output again in the end. So then let me show you also some nice things. There's a nice play button here. You can basically run the compartments. What it will actually do is basically pass along this command here to the actual output node. It does the same thing as if I go here and do update cache. For example, I can run this one here. And will output me the raw and pre-processed, and I don't even have to go into the pipeline itself and hunt down the node. And this is why it's very, very useful to set up these outputs always. So you can run the nodes from the compartments, so you can run parts of your pipelines without visiting actually the hierarchy of whatever is in there. Let's do this for the tissue and the glomeruli too. So I'm going to add here output by clicking just the plus button and then leaving it as the default for the glomeruli too. And then let's output some things directly from in here. And then maybe let's output the visualization and the measurements. So in the tissue, I'm dragging it here to the plus because I don't want to hunt down the RGB image. And call it the visualization. And then drag the thing that you want to go out to the plus. Then it snaps into place and turns green. You will see on the left-hand side, it will auto-select what is the type of data. And then you give it a name. And I'm going to press add. So then they're both out. Then I go to the glomeruli. There's also, again, this output node. And then I'm going to add the visualization. And add it in. And then from the measurements, I drag it into the plus. And then now the outputs are set up for both of them. And then this is nice because when someone else wants to run it and wants to run specific parts of your pipeline, they can just go to the compartments overview and then run it from here. So it's there. I have my measurements still here. I can read them directly from here. My visualization for the tissue, we have exactly the same thing.